everyone, I hope you will be fine. And so in this video we will discuss about different genetic diseases which occur either by mutations in protein coding genes or there is an alternation in protein coding genes other than mutations, like there may be structural variation in DNA sequence. In this video I will also tell you about Mendelian disorders and their transmission patterns. So let's get started. So what was the need to study about genes and what was the purpose of Human Genome Project? First of all, we should know about this. The Human Genome Project was actually an international research program whose goal was the complete mapping and understanding of all the genes of human beings. All our genes together are known as genome, and the knowledge that is obtained from this project is leading us to new ways of diagnosing, treating, and possibly preventing diseases. There are about 20 to 25,000 protein coding genes, and we can now sequence a human exome and is applicable as personalized medicine. And what is personalized medicine? And this is actually personalized is the treatment that is directed at the symptoms, and this treatment is adjusted depending upon the patient's phenotype. And we should understand these three terms before studying the genes: hereditary, familial, and the congenital. Hereditary are the factors that are transmitted from one's parents, and familial cases when there are more cases of disease in history of one's family and there is a susceptibility of developing disease, while congenital are those that are present at birth. So next is mutation in protein coding genes. Mutations actually refers to the permanent changes that occur in the DNA sequence, and that change may be in the germ cells or that may be in somatic cells. If their mutations are occurring in germ cells, they can be inherited to the next generation, and if their mutations are in the somatic cells, they are not the inheritable. And so basically there are three mutations that we should have a look upon them, like the point mutations, frameshift mutations, and trinucleotide repeat mutations. And the first one is point mutation, and that is actually when there is a substitution of single nucleotide base by a different base, like in the sickle cell anemia and the Tay-Sachs disease. In the sickle cell anemia, there is a deficiency of red blood cells that carry oxygen throughout our body, and there is a mutation in a beta globin chain of hemoglobin, and the glutamic acid is replaced by valine at the 6th position. We know that our red blood cells are round shaped, and the codon that is mutated in sickle cell anemia is the GAG, which is a normal codon. And that normal codon, and in its the complementary the state, there will be the CTC, and the glutamic the amino acid will be formed that is in the normal state. And sickle cell anemia is named because of its the sickle like the shape, and there is the change of the A to the T, and in its complementary there will be the A, and so new codon that will be the formed like in a sequence like the CAC, and that will form the valine, which is in a mutant state, and there will be the deficiency of the oxygen. And if we see patient the blood, and there will be red blood cells like in this the state sickle cell shape. What is the frameshift mutation? And frameshift the mutation and how this occurs, let's have a look on this example. And CAG, CCC, and ACT, these are the three ketones, and every keton codes for one amino acid, and like the CAG forms the glutamine, triple C forms the proline, and ACT forms the threonine. And how this frameshift mutation happens is that there is either there is the insertion or there is a deletion of one or two base pairs that alters the reading frame of DNA, the strand. For example, here there is the addition of this the nitrogenous the base, and so new codon will be TCC, and that will actually form the serine instead of the proline. And here the C that was the here will be the shifted to this position, and the next codon will be changed into CAC, that will form the histidine, and so new sequence of amino acid is changed, so protein product will be changed completely. And then what is the trinucleotide repeat mutations, and that are characterized by amplification of sequence of 3 nucleotides, and 3 nucleotides are seen in different cases in different positions more than one time. For example, in Fragile X syndrome, and we can see the expansion of CGG triplet nucleotide many times in MR1 gene. And now we'll move to alternation in non-coding RNAs. And non-coding RNAs, they are called so because they will not produce any of the proteins, and they have the important regulatory the functions. And examples of non-coding RNAs are small RNA molecules called the microRNAs, and there are the long non-coding RNAs that come in the category of the non-coding RNAs. And what are the major categories of genetic the diseases? And there are the 3 categories: Mendelian disorders, complex disorders, and chromosomal abnormalities. Mendelian disorders that are when one gene is affected, and they are the non-Mendelian disorders. They are the actually mutations of the single gene, and they are very common the conditions like the storage disease and inborn errors of the metabolism. When there is a role of the multiple the genes on a disease, and there is also influence of the environmental factors, then they are called the complex disorders like hypertension, diabetes, and allergy. And when there is a change in a number of the chromosomes than a normal, and or we can say there is a change in the structure of the chromosomes, then they come in category of the chromosomal abnormalities. So what are the Mendelian disorders? And I have already told you that these diseases caused by the single gene effect, and they show 3 patterns of inheritance like autosomal dominant, autosomal recessive, or X-linked. And there we should know about that a single gene may show a phenotypic expressions, or the mutations at several different loci may produce the same expression. For example, if we see Marfan syndrome, the disease in which there is a mutation in the gene that codes fibrillin, and may have, but that gene may have the widespread effects involving affecting our skeletal, eyes, and cardiovascular system. And its extent, its the expression, and the severity of the expression may be influenced by other genetic loci, and which are called the modifier genes. And there are the 3 patterns of autosomal dominant, autosomal recessive inheritance, and X-linked disorders. And we do not discuss about Y-linked the disorders because no any Mendelian disorder have ever been reported in Y category. And what are the patterns of inheritance of autosomal dominant inheritance? And they are manifested in mainly heterozygous state, and here the both males and females are affected, and both sexes can transmit this condition. And what are the major features of this inheritance? That there is a 50% chance of the children of being affected. And so there may be some cases that the patients do not have affected parents because they own the disorder by mutations involving either the egg or the sperm. In many of the cases, the symptoms do not appear in adulthood, and there is a 50% reduction in a normal gene product. And what happens is that, for example, if we take an example of the collagen molecule, and which is a trimer, means that they have the 3 collagen molecule chains. And if there is a 1 mutant chain, and that will reduce the assembly of remaining the chains, producing a marked deficiency of the collagen. And examples are the myotonic muscular dystrophy and Huntington disease. If the father is the affected, and father has the affected the gene affected a mutant, affected mutant the gene, and mother is the unaffected, and that the gene will go into the gametes formation, and the next will go into the next generation. And so two of his children will be affected and 2 will be unaffected. There is a 50% chance of the children being affected in the autosomal dominant inheritance. If we talk about disorders of autosomal recessive inheritance, and they are manifested in homozygous state, and there is a 20% chance of being affected in progeny. And new mutations for these disorders may occur, but they rarely happen in this case, and after several generations an affected person may born. In this case, enzymes are also affected by the mutations, but in heterozygous equal amount of the normal and the defective enzymes are synthesized. So the cells that have half of the complement of the enzymes, they function normally. And example of autosomal recessive inheritance include sickle cell anemia and the cystic fibrosis. If the mother and the father both are the carrier of the mutant gene, then the one child will be the affected, one will be the unaffected the child, and while the two will be the carrier, the children. And what are the X-linked disorders? X-linked disorders are mostly X-linked recessive, and they show the heterozygous the females actually rarely express the full phenotypic the change because they have the paired normal allele. Like autosomal linked recessive the inheritance, a woman will be affected if the two alleles are both mutant. Women are affected if the women are affected, they are both copies of the disease allele must have the defective the gene, and if woman is affected, all of their sons will be affected and all the daughters will be unaffected carriers. If a male is affected, actually male does not transmit this disorder to the sons, but all the daughters are the carriers, and sons of heterozygous the woman have one chance in two of receiving the mutant gene. And here we can see if the father has a mutant gene, then no one will be affected in this in the next generation, but they will be the carriers. If the mother has the mutant gene on the one side, and then this one son will be the affected, and other no will be affected. And I hope you like this video, and if you like my video, and give it the thumbs up, and I will try to improve my videos next time, and my next video will be on the syndromes. And if you really like my video, please give it a thumbs up, and if you want to see more videos like this in future, hit that subscribe the button. If you want to see me cover another topics, leave a comment below. And alright, well, that's all I have for you today, and I hope you enjoy this video. I hope to see you next time. Goodbye.